So this is a introduction to gelatin as part of the lab we're going to do on the elasticity of jello and gelatin. First thing you should know is that gelatin is a protein and it's derived from animals. So jello, the jello that you eat, that you know and love, is actually derived from animals. The protein is collagen, and collagen is a protein that consists of three separate chains, all attracted together in a helix structure. So they wind around each other. And that picture is shown here. Collagen makes up a lot of the connective tissue, the ligaments, the tendons in animals. Also makes up the cartilage, the vertebrae cartilage between different vertebrae, and it's also a component of skin. So it's the connective tissue under the skin, the layer of skin cells. So you have the dead skin cells that are on the top that are on the outside, then a layer of live skin cells that grow, and you have hair follicles mixed in with those. And then below that is a collagen layer, and collagen also makes up the walls of blood vessels. So collagen is widely used in the body in animals and people as connective tissue to hold things together. One of the ways that we get collagen one of the ways that we collect collagen is when you see collagen when you cook meat. When you at the bottom of a roasting pan, after the roast has been cooked, you'll see after it's cooled, you'll see the gel kind of congeal, and that's collagen. And if you take that and purify it and get out all of the other salt and other meat proteins and juices, and you just got the collagen left, that's what you're going to use for Jello. So, like other proteins, collagen will denature to form a gel, and you denature it by heating it, and it breaks up the strands, and then they reform into a web that can absorb water. So you can imagine, for example, having balls of yarn that you then unstring and then throw at each other and then try and pull and sort them out, and then they just start to tangle up and form this web, and then each individual protein starts to cross-link not to in an organized structure but to a random thread of another protein, and they form a web. These webs can form cells that can then fill with water. So most gels are simply 1 or 2% of the polymers by weight, and then mostly water. And so that's why water is such an important component when you're making gelatin. And so here's some microscopic images of different gels at different temperatures, and you can see the web that they can form, and the cells that would absorb the water and hold and because of that become squishy and springy.
What are you up to today?
I'm looking at gelatin crystals on the microscope here. We got a picture of a microscope picture. And I'm going to try and bloom them, that is, get them to absorb some water.
Cool.
Let's see if I can videotape that. These are microscopic pictures of gelatin crystals under a microscope taken with my cell phone with a strong magnifying lens. Then you're also seeing the gelatin absorbing water here. So in this video in the lower right, you saw one of the crystals absorb the water, become very translucent as it absorbed the water because its properties became more and more water-like. This is a second video showing another set of gelatin crystals absorbing water and blooming up, getting bigger, and becoming more and more water-like. As with other materials, you can measure the elasticity. We will measure the elasticity from the data that I will give you. You can calculate the elasticity, and then from that, calculate the connection spacing, where the tangles of the different polymers are, and how far apart those tangles are on average. So we can use the elasticity equation or elasticity relationship that we have, that it's along with being able to calculate it from the stress and strain, you can calculate it theoretically from kBt over the cube of the distance between the different connections. For a gel to melt, most gels melt at about 300 degrees Kelvin, so about 30 degrees Celsius. So, somewhere between room temperature and body temperature. And so from that, we see that the interaction energy is about 4 times 10 to the minus 21 joules. So we should be able to, once we get the elasticity from the stress and strain, you can plug that in and calculate the connection spacing for each of the different types of gels that will form. So that's a brief introduction to gels and gelatin, and you should go on to view the other 2 videos regarding the lab.